Key area, structure and replication. 1. What is differentiation? Differentiation is when unspecialized cells, e.g. stem cells, have genes switched on so that they can perform a specific function or role within the human body. 2. Give an example of a differentiated cell type. Examples of differentiated cell types include nerve cells, red blood cells, formed in bone marrow, and cardiac muscle cells. 3. What method do somatic cells use to divide? Somatic cells divide by mitosis. 4. What method do germline cells use to divide? Germline cells divide by meiosis. 5. How would we describe the chromosome complement of somatic cells after cell division? Somatic cells would be described as being diploid after cell division. 6. How would we describe the chromosome complement of germline cells after cell division? Germline cells would be described as being haploid after cell division. 7. If a mutation occurs in a somatic cell line, what impact would it have? A mutation in the somatic cell line would directly affect the individual. 8. If a mutation occurs in the germline cell line, what impact would it have? A mutation in the germline cell line would have an impact on the offspring. 9. What is a benign tumor? A benign tumor is an uncontrolled growth of cells that remains in one location. 10. What is a malignant tumor? 11. A malignant tumor is an uncontrolled growth of cells, e.g. cancer, that break through the cell membrane and detach, spreading through the body. 12. Identify the base pairing in DNA. In DNA, adenine pairs with thymine and cytosine pairs with guanine. 13. What are the differences in bases between DNA and RNA? In RNA, thymine is replaced with uracil. 14. What type of bonding holds the complementary base pairs together? Hydrogen bonding holds the complementary base pairs together. It is a weak bond and when broken allows the DNA to unzip. 15. What type of bonding holds the phosphate and sugar together in a DNA strand? A phosphodiester bond holds the sugar and phosphate together. This is a strong bond and helps maintain and support the backbone of the structure. 16. What type of sugar is present in DNA molecules? Deoxyribose sugar is present in DNA molecules. 17. What type of sugar is present in RNA molecules? Ribose sugar is present in RNA molecules. 18. DNA is described as antiparallel. What does this mean? One strand of the DNA runs in the 5' to 3 direction and the other strand runs in the 3 to 5 direction. 19. How are fragments made in the lagging strand of DNA? Fragments are made at the lagging strand of DNA due to the primers only being able to anneal, join, at the replication fork. More primers are added as the DNA strand continues to unzip.